Hello students, welcome back to my YouTube channel. This is Dr. Sumayed from L Biotutor. In this video, I am going to talk about polypeptide synthesis. So, what is polypeptide synthesis? This is called, simply saying, protein synthesis. Protein synthesis in the cell. This process is starting in nucleus, and you can divide this into two simultaneous processes. That is transcription and translation. Transcription means copying of a sequence of DNA to a mRNA. This is happened inside the nucleus. So the genetic information in the DNA will convert it into mRNA. DNA information, that DNA sequence go into mRNA. So you will produce a mRNA. The process of copying sequence of DNA to mRNA is called, that is, transcription. The next process is translation. The second part of the protein synthesis is called translation. So in this part of the process, convert the information in the mRNA to a sequence of amino acid. So sequence of amino acid with the polypeptide bond, you call it a polypeptide. With the peptide bond, it is polypeptide. So it is protein. So as the ultimate result, you have a protein. As this figure is there, so DNA, copying of DNA, as we learned in the previous video, I have talked in the previous video that about the replication. It is in my YouTube channel. You can watch it. So DNA copying is replication. DNA sequence going to RNA is transcription. And mRNA, that mRNA converting to protein, that mRNA give rise to protein is called translation. And the another process is there, which is with the mRNA, with using of mRNA, prepare the DNA sequence, that is reverse transcription. This is happening in with the reverse transcriptase enzyme in some kind of virus. That we will discuss in microbiology lesson. So as summary, so DNA convert into mRNA, that is transcription, and mRNA convert into amino acid, that is translation, is the two main process of protein synthesis. Now we will discuss this as this with a description. This transcription process, you can divide it into three parts: initiation, elongation, and termination. Initiation process, you have something called promoter sequence. In the DNA, you have a specific sequence of the DNA, so that is called promoter sequence. From that place, you are starting the mRNA synthesis. mRNA synthesis, that is transcription for the protein synthesis. We have complementary true strand in the DNA, but one strand acts as a template. Although DNA replication, you have used both DNA strand, dear student. In transcription, it is only one DNA strand is used as a template. This is very important. This one you can take it as a MCQ point. So one strand used as a template. And the enzyme RNA polymerase is the enzyme very important in this initiation process. We can remember that in DNA replication, for the unwinding of the DNA, we have to use helicase. In transcription, the initiation process, there's no helicase involved. That helicase activity is done by RNA polymerase. So unwind of the two DNA strand and beginning of the transcription is starting with RNA polymerase. Next is the elongation. With the elongation process, RNA polymerase add complementary ribonucleotides. Previously, we know that DNA deoxyribonucleotide added, here ribonucleotide added. So this is against the template, DNA template, that is 5' to 3'. This elongation process is continued up to termination site. There's a specific site called termination site of the DNA. So up to that point, this RNA polymerization is continuous. While the mRNA is processing, mRNA is synthesizing two strands of the DNA rewind at the other end of the DNA. So in one side, unwinding and RNA is processing, RNA synthesis with using of one DNA strand. At the end, it is winding again. The termination, that is the third part of the transcription. So transcription we divided into three parts: initiation, elongation, and termination. As the third part of the termination, we can see some difference in prokaryote and eukaryote. Prokaryote polymerization continues until the termination sequence, and the termination sequence RNA polymerase fall offs and mRNA. You can use the mRNA. But the process is somewhat different in eukaryote. You have the new pre-mRNA that is on the mRNA, so it's called a pre-mRNA. So you have some mRNA processing, and after this mRNA processes, mature RNA will leave the cytoplasm from the nucleus. Now we know that mRNA is produced at the nucleus with use of the DNA. This mRNA will go in through the nuclear membrane to the cytosol. That is why you call it as a mRNA. It's a using as a messenger. So it will act as a messenger from DNA to cytosol. That is mRNA. We call it mRNA. Right. So translation. In the translation process, your mRNA converted into amino acid sequence. This one also you can divide into three parts, three sub processes: initiation, elongation, and termination. The translation initiation, we will have important point here. We know that in cells we have something called ribosome. Ribosome has two subunit, that is small subunit and large subunit. Small subunit of ribosome bind to mRNA. The mRNA which was coming from the nucleus, which has been transcribed with the DNA, and this small subunit bind to mRNA. And the other one is the initiator tRNA. There's an initial tRNA, and also with these three large subunit of the ribosome combine to form the functional ribosome. So this is you call translation initiation complex. Okay. Translation initiation complex. So ribosomal subunits, mRNA, and initiator tRNA. All these three call translation initiation complex. Two ribosomal subunits, mRNA and initiator tRNA. So the initiator tRNA is very important. We know that initiation is starting with initiator tRNA. So the mRNA with AUG codon. This is the mRNA AUG site, that is you call start codon. This is aligned with P site of the ribosome. So that is happening at the initiation of translation. You can see it like this. So mRNA, two parts of the ribosome subunit, tRNA. So these are the parts of the translation initiation complex. So AUG, that is start codon. So the start codon, anticodon is, as you know, that anticodon is UAC. That tRNA is aligned with the P site of the ribosome. There is a middle site. There are three sites of the ribosome: E site, P site, and A site. So this tRNA will, as in this figure, they are with the initiation part. So lower left side. So it is tRNA. tRNA. Right. So that is initiation. Elongation process is also very important. Now we have A site is empty. This empty A site along with the next codon. So we know that AUG codon is they are aligned with the tRNA, and A site, the next A site is aligned with next codon. So for that place, next RNA will come to that site, that is A site. When tRNA is coming, with the tRNA, we know that amino acid is coming. That is why you call it as a tRNA, transport RNA. tRNA coming with amino acid. So the amino acid of tRNA, the previous one and the next one is coming together. And what happened here? Peptide bond is made with this through amino acid. So you can see it in the figure. So peptide bond carboxyl group of P side and amino group of A side will form the peptide bond. And rRNA, that subunit of the ribosome RNA, catalyze this reaction. So you have the catalytic action of the reaction by the ribosomal RNA. And next thing is translocation of the mRNA with the using of GTP. What happened here? So mRNA moves codon by a codon. mRNA moves one codon. At that moment, the tRNA which is there at the P site now come to the E site. That is called exit site. And tRNA that was there in the A site come to the P site, and new codon there. So new tRNA will come with a new amino acid. So mRNA moves codon by codon. At that moment, A site tRNA moves to the P site. So the tRNA which was there with the amino acid, while peptide bond is forming, this connection with the amino acid is not longer there with the tRNA. So tRNA will release from the E site to the cytosol. Release tRNA. So it is released to the cytosol, and the A site will align with next codon, and this process is continuing. So this will use GTP as a, this is a good that it will use energy, something called anabolic process. So you can use the, sometimes we use ATP, here we use GTP as the energy source for the protein synthesis at this place. So termination, the mRNA aligning with the stop codon. So mRNA align with the stop codon at the A site. A site align with the stop codon in the mRNA. So there are three stop codon. So AUG is the start codon. So normally we call that August, AUG, August. So you can remember like that, August, AUG. So that is start codon. Stop codon, there are three stop codon, students. Okay, so it is UAG, UGA, UAG. All three are starting with U, and UAG, UGA, AG, 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 GA. So you can remember that one as UAG, UGA, UAA, because these are important. So these, please remember these three and the start codon. Other codon, not necessary to remember what the amino acid is given, but remember these things: stop codon and start codon. Start codon, it is you given the methionine, that amino acid. When you have the stop codon there, there's no tRNA for that stop codon. So what happened there? Rather than a tRNA, release factor is bind there. So because with the tRNA, we have the amino acid sequences coming with the peptide bond, and while you have the release factor there, so you don't have the amino acid. So that continuation is stopped at that moment, and amino acid sequence will release to the cytosol. So this peptide chain. So that's you call translation assemble fall apart because of the release of the polypeptide chain. So this simply saying this is the protein synthesis. You have two processes, transcription and translation. Let's see it again. Translation initiation as the large unit and small unit of ribosome, mRNA and tRNA, initiated tRNA. So you have the translation initiation complex and elongation happening with the using of GTP and termination with the stop codon. One of stop codon. There are three stop codon. So you can see clearly the initiation, elongation, and termination process. Right. Not only one ribosome, for one same mRNA can bind with many ribosomes. So this will increase the efficiency of the protein synthesis. You call it a polysome or polyribosome. mRNA with multiple ribosome, you call it a polysome or polyribosome. And this will increase the rate of translation. So it is important for the protein synthesis. So you will have many protein chains with using what? One mRNA actually. With one mRNA you have an amino acid sequence. It's same, equal, identical sequence. So then what happened to this protein? We say that this is protein synthesis for the using of amino acids. So you make the protein. So what happened to this protein? So you call it a fate of the protein. So naval synthesis peptide, you call it a primary structure. Now we know that there are four structure as we learned in the second lesson of the syllabus. We have discussed in the second lesson. I have discussed that one also in the video. So primary structure, secondary structure, tertiary structure, and coordinate structure. That is due to protein folding. So primary structure is there, and with the protein folding, you will have secondary or tertiary structures. And some polypeptide chains, you will have signal peptide. This peptide will guide you to location of the cell, where to go, where should you stop, where to go to the, what is the function of the protein that is given by that signal peptide. And sometimes these peptide will be secreted. Some protein will be secreted. So these extra pieces will removed enzymatically. And also we understand that every protein starts in synthesis with AUG, that is called methionine. So that methionine part should be removed. That will happen as post translation modification. It's a chemical modification, and sometimes your protein will be cut into pieces. Sometimes these pieces may be combined. Those are post translation modification. Also, you will have sometimes this protein will combine with sugar. You call it glycoprotein. Protein will combine with lipids. This is called lipoprotein. Sometimes protein combine with phosphate groups. So phosphorylated proteins. These are the different types of protein you will have. And this is the protein synthesis. And also there are protein degradation. You call it a selective degradation. So inside the cell, the amount of protein will depend on the synthesis and degradation, production of the protein and destroying of degradation of the protein. This will control by the cell. And damaged proteins, also regulatory proteins after their function. So you have to degrade those things. So you call them selective degradation of protein, damaged protein and regulatory proteins. You can visit my website. So you can watch my videos on YouTube AL Biotutor. Thank you for the listening. Let's meet up in the next video. Thank you.